Welcome to Alliance for Science Live. I'm your host, Chris Knight, based at Cornell University in New York. I'm joined today by Professor Ray Dixon. He's from the John Innes Center in the UK. And today we're going to be talking about engineering biological nitrogen fixation. So thanks so much, Ray Dixon, for joining us on the program.
Well, thank you, Chris, for the introduction, and thank you for giving me the opportunity to be here.
That's great, and we're happy to have you. So for folks who don't know anything about biological nitrogen fixation, uh, Professor Dixon has been working to understand unique relationships between crops and nitrogen-fixing bacteria. These bacteria live in the soil. They're able to convert nitrogen that's existing in the atmosphere into a form that can be taken up by plants. And some bacteria might be able to share more than others. We'll learn about that. But essentially, legume crops like alfalfa, clover, beans, lentils, there are leguminous trees like the locusts. These trees have the ability to create a symbiotic relationship with these bacteria if they're present in the soil. And this is a benefit that both small and large-scale farmers across the world have been using for millennia, whether they have known about the exact microbial process or not, but legumes are present in farming systems all over the world. So we're kind of thinking about, hey, if the symbiosis between legumes and nitrogen-fixing bacteria could be engineered to work with other crops like cereals, or if it could be engineered to work in a more beneficial way to growers, that could provide farmers and consumers and the environment with huge positive benefits. Now, these processes are complex, and scientists don't know everything about how they work, but that's why we have people researching it to try to figure this stuff out. So, Professor Dixon, I'm wondering if you could give us a little bit of background on biological nitrogen fixation and why is it important, and if you could kind of explain it in a way that maybe a non-scientist can understand what's the problem that your research is trying to solve.
Well, Chris, you've already given a little bit of introduction to this, which I think was very helpful. Nitrogen is, of course, an essential component of life. It's a key element in many biomolecules, for example, being DNA and proteins. But plants and humans can only assimilate a certain form of nitrogen, which we call fixed nitrogen. And this is the nitrogen element combined with other elements, particularly hydrogen to form ammonia, or nitrate, which is an oxidized form of nitrogen. Now, we're fortunate in some ways that nitrogen as an element is extremely plentiful in the biosphere, since our atmosphere consists of around nearly 80% nitrogen and 20% oxygen. But nitrogen gas, as Chris has already mentioned this gas can only be fixed into ammonia by bacteria and another group of simple organisms called archaea. So the process of biological nitrogen fixation involves the conversion of nitrogen gas to ammonia by these bacteria, thus producing a form of nitrogen that can be assimilated by plants and animals, and Chris introduced that to you. And he also emphasized that the process is incredibly important, since nitrogen is a critical element for crop growth. So in our research, what we're trying to do is to understand precisely how this process works, how it is regulated in response to the environment, and how we might manipulate nitrogen fixation to improve agricultural productivity and develop a more sustainable agriculture.
And so, to follow up on that, one thing I want to, another point I want to lay out there is the fact that this is a process that farmers have been utilizing and benefiting from for a long time. People have realized that legumes don't really need as much fertilizer, or don't need fertilizer applied to it compared to other crops. People have kind of been using legumes as a way to as a part of their crop rotation for a long time, and we're kind of just recently uncovering these secrets about why that exactly works. But so, do you think that there would be a demand for um, figuring out more about how this process works, just based on how widespread legume growing is in the world?
Well, there's a tremendous amount of fundamental science going on in trying to understand how the legume nodule symbiosis works. So it's a very sophisticated symbiosis with nitrogen-fixing bacteria, the emphasis being that it's the bacteria who fix the nitrogen. And in the context of legumes, these symbiotic bacteria are collectively known as rhizobia. They engage with the plant. In fact, they infect the plant, which in response to the appearance of the bacteria, the plants develop structures known as nodules on the legume roots. And within these root nodules, the symbiotic bacteria develop into a type of organelle that supplies nitrogen to the crop in exchange for carbon provided by the plant. So this exchange of nutrients is extremely important, since biological nitrogen fixation is an energy demanding process. And it is the carbon from the plant that supplies the energy needed to support nitrogen fixation. So the legume symbiosis is very efficient, and it can supply all the nitrogen demands of the crop. And it's therefore a prototype for engineering nitrogen fixation in the future. So that, I guess, is what we'll be following up on today. But I shall emphasize when we get around to it, Chris, that actually transferring the legume symbiosis to other crops, for example, cereals, is not the only way that we can manipulate nitrogen fixation. So I'll explain that, I think, a little bit further on.
That's great. That's great. So maybe you could just clarify a little bit. What is the root nodule? What are these nodules that you were talking about?
Well, if you've ever worked with legumes, and I guess many of you haven't, if you just pull them all out of the soil and you look at their roots, you will see that they've got these sometimes quite big bumps. It depends upon what kind of legume you're looking at. But you'll see these structures on the roots, which we call nodules. And these are the sites of nitrogen fixation. So within these nodules, the symbiotic bacteria have developed, so they developed inside the nodule, and they're committed, if you like, in that situation to fix nitrogen. That is their sole purpose in there. So the nodule is providing a little nitrogen fixing factory, if you like, on the roots of the legume.
That's really interesting. And then how does this bacteria get into the plant? Is it just kind of free existing in the soil already, and it just kind of finds a good plant and makes this match? Or what if a field doesn't have this bacteria present in it already, and you plant some legumes? Will you be able to get this effect? Where does this bacteria come from?
Well, that's a very interesting question. I mean, it depends upon the location and the soil type. So in some areas of the world, these symbiotic bacteria are freely available in the soil. And so all you have to do is to plant the legumes, and it works very well, because the the symbiotic bacteria is already there. In other parts of the world, you have to inoculate. So that means that you have to provide the bacteria to ensure that the symbiosis is successful.
Yeah, and then maybe this is a good time to mention the soil inoculants, because there are um, a number of products kind of coming on the market. It's kind of an emerging trend of products that claim to have this type of bacteria in it, and you could just inoculate your soil with it. And so, do you think that that's kind of a worthwhile approach, and how do you differentiate what you're doing to those kind of processes?
Right. Well, I'd like to point out here that we've been talking about symbiotic nitrogen fixation. We've been talking about symbiotic nitrogen fixing bacteria. But there are a lot of nitrogen fixing bacteria in soil. And they don't engage with legumes because legumes recognize a certain type of bacterium, as I mentioned, called rhizobia. But there are lots of different nitrogen fixing bacteria. They're very diverse. We find nitrogen fixation through many branches of the bacteria, and indeed these other organisms called archaea, and they exist out there in soils. Now, some of those bacteria, they make a contribution as well to nitrogen fixation. It's not only legumes, it's those bacteria. And so they contribute to the nitrogen requirement of cereals. So they don't contribute necessarily to legumes, but now we're talking about cereals. And if we're going to feed the world, we're interested in improving the nitrogen requirements of cereals. So these bacteria can enhance plant growth, and some can enter the plants to provide a more intimate association. We call these endophytes. And there are various estimates of how important these associative nitrogen-fixing bacteria can be to the growth of cereals. And one estimate has reckoned that it could be up to 50% of the N requirement. So this isn't as efficient as the legume symbiosis, but nevertheless, it's providing quite a reasonable contribution. But in this situation, because it's only providing 50% of the nitrogen requirement, some fertilizer has to be added. So part of the technology that we're interested in is developing this in some way. And I can talk, if you like, of the 3 different ways we are thinking about engineering these systems. But what has happened traditionally is that scientists have isolated many nitrogen-fixing bacteria that associate with crops. And as I've tried to emphasize, these are not necessarily the rhizobia, they're different bacteria. And what they do with them is they test them in the lab for plant growth promotion. And then they select the best ones, which can be used as inoculants for crops. And there are many biofertilizer companies throughout the world that are preparing inoculant formulations and selling these to farmers. Now, as I've mentioned, this technology is not as robust as the legume symbiosis. And one issue is that you find year-to-year variations in yield that are experienced by farmers. But the inoculants are far cheaper than nitrogen fertilizers, so they provide cost savings for farmers. But the issue is that this technology is influenced by the genotype of the plants, the climate, the soil type, and it appears to work better in some locations compared with others. In Brazil, for example, inoculants are widely used by farmers and are regarded as being relatively reliable, whereas in other nations, inoculants are not widely used, probably because chemical fertilizers provide more consistent yields. and farmers probably can afford chemical fertilizers. I should also point out that in the world, there is a dichotomy in terms of nitrogen availability. I'm talking now about chemical fertilizer availability, in the sense that developed nations can afford to use synthetic fertilizers, whereas developing nations often cannot. And for example, in sub-Saharan Africa, farmers can't afford to use synthetic fertilizers quite often. So these inoculant technologies can be quite useful if they work. And as I've emphasized, they don't always work, and that is an issue.
So you really need to provide that match between the genotype of the plant and the environment and the soil type and the right concoction of the bacteria that you need in order to get that reliable result. And so there are a lot of factors at play. And but okay, so moving on, I'm curious about then kind of how did you kind of make your way into this kind of research, and then let's talk more about the kind of inroads that you're making in the engineering side. You mentioned that you kind of have three potential ways that this biological nitrogen fixation process could be engineered. So maybe if you could talk about how you made your way into this field, what excited you about it, and then what are these three different options that you could present to us?
Okay. Well, I've worked on this problem for more than 50 years now, and I've seen some amazing changes during this time. Why did I get interested in it? I was an undergraduate student at the University of Reading in the UK, and we used to have tutorials, and we used to have tutorial topics in which each of us would be invited to speak on that topic. And I just happened to be reading something on nitrogen fixation and got interested in it. I was really fascinated by it. And I had several opportunities for doing doctoral research at various universities. But I was particularly attracted by the University of Sussex in the UK because the government at that time had set up what they called a unit, a small group of scientists brought together to investigate a given problem. And this unit was brought together to investigate nitrogen fixation. And we were a group of chemists and biologists working together on this problem. And it was fascinating as a postgraduate student. What was even more fascinating was I was suddenly presented with a completely new challenge in research when I was just thrown in at the deep end, actually, on this issue, because I thought I was happy with biochemistry. And in those days, we were developing biochemistry, trying to understand how this process works, because you need a special enzyme to do this process. But nevertheless, the director at that time, who was my PhD supervisor, said to me, "Ray, how do you fancy doing some genetics?" And I was kind of horrified at the time because my genetic training as an undergraduate had not been that good. And really, nobody knew anything about the genetics of nitrogen fixation. And in those days, we didn't, of course, have the technologies available to us that we have now. There was no DNA cloning, for example. So in those initial days, the only thing that we had available to us was to inactivate genes. So we could do that in bacteria. So we could ask the question, can we make mutants that don't fix nitrogen? And the other thing we could do is to use natural DNA exchanges to transfer genes from some organisms to another organism. So this occurs naturally. It only occurs between relatively related organisms. And I was very lucky in my PhD because I managed to create a genetic system in which we could transfer the nitrogen fixation genes from one nitrogen fixing bacteria to E. coli. And E. coli is the model, if you like, for bacteria. It's been extensively studied. And so we created the first new engineered nitrogen fixing organism without using DNA cloning. So that was very successful. And from that time on, we could start asking the big questions. How many genes do we need in this process? How does it work? What genes encode the enzymes that do this process? That kind of fundamental question. How is this system regulated? Because we know it's regulated. If these cells have got too much nitrogen outside them, the process is switched off. And that's true out there in the field. If you have got too much ammonia, if you throw a nitrogen fertilizer onto a legume, you'll kill nitrogen fixation, for example. And after the advent of DNA cloning, we could clone these genes. And that gene cloning allowed us to look if other organisms had similar genes, and eventually one could clone the genes for nitrogen fixation out of the bacteria that interact with legumes, for example. Does that kind of answer your question? That give you a flavour for how I got into it?
Yeah, and then continuing on. So the three approaches that you hinted at before, could you talk about those?
Yes, certainly. Let's talk about those. That's kind of exciting. I mean, that's what continues to excite me these days. And we don't really know which of these will be successful at the end of the day. But let's start first by thinking about the kind of things I said about inoculants. So I've already said that the legume symbiosis is really successful. But there are these inoculants that we've been talking about. And the aim of this kind of area is to improve this. And one of the ways of improving these kind of associations is to make sure that the bacteria actually deliver their nitrogen to the plant. One of the issues here is that because these bacteria are free living, they fix nitrogen for themselves. They want that nitrogen for themselves. They're not particularly altruistic. And so what happens with crops is that they will get nitrogen from these bacteria, but probably only when they start to decompose. When the bacteria grow well in the soil on gaseous nitrogen, their biomass increases, but they will die and release that nitrogen, and the crop can benefit from that. So one of the aims of the engineering is to change that so the bacteria give up their ammonia for the benefit of the plant. So we're making it towards a more, if you like, symbiotic process.
Yeah, so kind of engineering them to be able to share a little bit more.
That's right.
Of the meal that they're making for themselves. Get a little bit for themselves and give a little bit away as opposed to how they've evolved to keep all of it to themselves.
That's absolutely correct, Chris. Yes, that's the way. That's one of the ways we see forward. And we can do this. So, as you say, we can make them give a bit of their meal to the plant. The slight disadvantage, and and by the way, there are experiments in the globe which show that this does work. So you get increases in yield when you do this. There are not many studies on it yet, but there are some studies, and even in the US, for example, there are companies now, and they have products based on this kind of technology, and farmers are using them, even in the US. There is a slight disadvantage in this, in the sense that you're giving something away, and if you give too much of it away, you don't become too healthy, you kind of starve. And if you're giving too much away, then you're not competitive. So everything out there around you can grab that stuff, for example. So they can say, "Oh, thanks very much, I'll have that," rather than the plant. So
this could be other organisms in the soil, or
other bacteria in the
weeds.
soil will say, "Yes, thank you very much, I'll have that." So what you need to do there is to try to increase the specificity of the association between the crop and the bacteria. So we're building that kind of interaction in here. So one of the ways of doing that is making sure those bacteria don't give away their stuff, don't give away their meal, don't give away their nitrogen until they're right there in the right place in association with the crop. So that means that you then have to engineer the plant to provide some signal to tell those bacteria to release their nitrogen when they're ready. And that technology is already being developed. So people now have managed to do that so that signals can come from the crop plant to the microbe. It isn't quite complete yet. They haven't completed the cycle, so they haven't got it to the state where the crop is actually telling the bug to fix nitrogen. But nevertheless, they have got signaling pathways working. So we are on our way towards creating this synthetic symbiosis. And one of the other things we can think about is maybe making sure the bacteria have enough food, if you like, because we need to exchange the nutrients again. We have to give away our nitrogen. The bacteria have to give their nitrogen to the plant, but the plant in exchange could give more carbon so that there's more energy for the process. And therefore that will increase the efficiency of the synthetic, what we call synthetic symbiosis. So that's the kind of number one, if you like, or the first thing that I was talking about, I want to talk about in terms of engineering. Now, the second one is the one that I think you perhaps, or people might be more familiar with, and I'm not doing this, engaging in this research myself, but many others are. And that is what we've mentioned already, is to engineer the legume nodule symbiosis on cereals. But this is long-term, and it's long-term because it's a very tremendously, we've mentioned, sophisticated symbiosis between symbiotic bacteria and legumes. It's very sophisticated. And we have these nodules and so on. So if you want to engineer this into cereals, it's going to be quite a challenge. And there are 4 steps, basically, that you need to do this. And the first thing is you have to engineer the talking together, the signaling. So that means that if you're engineering this into a cereal, the cereal has to say, yes, I've found the rhizobium bacterium. There's a rhizobium out there in the soil, and I'm going to respond to that and make a nodule. So you have to get them talking together, if you like, exchanging signals. Then you have to engineer this structure on the root. So you have to get the genes in there, which will allow you to engineer this nodule on the root of those organisms. And then you have to engineer a way in which the bacteria can enter the plants in a safe way and get into the nodule. And there are some issues here because this has to be a very specific interaction. You don't want pathogens to get into your plant. And so again, you have to be very careful about engineering that infection process. And finally, you have to engineer the right environment for nitrogen fixation. And that means, I haven't mentioned this, but the system is very oxygen sensitive. So you have to find a way of protecting the enzyme from oxygen damage. So it's a very ambitious objective, but people are getting there. So already it's known that there are shared signaling pathways, for example, in legumes and cereals. And so one of the first steps in the process is already beginning to be achieved. So the third way I'm going to talk about is forgetting about the bacteria altogether. Let's forget about them. Let's instead do some real genetic engineering. Let's put the genes for nitrogen fixation in the plant instead. No bacteria, just genetically modify the plants. Now, this is also horrifically complicated for a huge number of reasons, but one of the reasons I've already mentioned is that the enzyme, I use terminology here, I'm sorry, this enzyme is called nitrogenase. It's very sensitive to oxygen. So we have to find a location in the plant which will protect it. The legumes have already done that, they've invented the nodule, but what can we do with the existing features present in a plant? And the proposal is that we put not the genes, but make sure the proteins go into organelles. And there are two types of organelles in plants we tend to think about. These are mitochondria, and these are the energy generating organelles in plants. And there are chloroplasts. And chloroplasts are the site of photosynthesis. Now, mitochondria should be quite a good environment because they have active respiration. This means they respire oxygen very fast. And that means that the internal cytoplasm, what we call the matrix of the mitochondria, should be oxygen deficient. And that turns out to be true because some scientists recently in Spain showed that the most oxygen sensitive component of nitrogenase could be made in an active form in yeast mitochondria. Yeast are easier to work with than plants, so they did it with yeast. It is being done with plants, and it is being done with plants, and it is working in plants, but they showed it first in yeast. So that means that mitochondria are a good environment for nitrogen fixation in plants. The other possibility I mentioned is chloroplasts. And if you think about chloroplasts, that's really paradoxical because chloroplasts are the organelles that do photosynthesis. So they're going to produce oxygen. But we know from our knowledge of bacteria that they've solved this problem. So there are groups of bacteria called cyanobacteria, which actually are photosynthetic and they fix nitrogen. And they do this in a variety of different ways, but one of the ways they do that is to fix nitrogen in the dark. And obviously perform photosynthesis in the light. So the two processes don't go on at the same time, and these cyanobacteria have methods of protecting the nitrogenase from oxygen damage. So that is another possibility. And the other big issue with all this kind of engineering is the number of genes that you need to introduce. And I mentioned that the number of genes is quite large. So in the sort of work that we've been doing, it's about 17 genes. And that's a large number of genes to put in there. And the other problem is that you have to be very precise in the engineering, because the system is very, very balanced. You have to have the right balance of components being expressed in the plant. And that's a bit of a challenge, because one of the things of doing this kind of engineering which makes it difficult is that bacteria, where these genes come from, of course, they have a different way of expressing their proteins than plants or animals. And to do this, what we call synthetic biology, that means you have to manipulate all the genes and all what we call the expression systems to actually get the right level of these proteins in the crop. And that's very, very complicated. So we have the problem of complexity, and we have the problem of oxygen sensitivity, but the oxygen sensitivity problem has been resolved. But with all these challenges, bottlenecks can arise, and you're never quite sure when you're finally going to get there. So that's my three, if you like, ideas. They're not mine entirely. There are many, many people working on this in the world.
Of course. But wow, thank you so much for that explanation. So you covered these three main kind of theories of how this could be done. One is trying to engineer the bacteria so that they share a little bit more of their meal with the plants that are around, and the second way of looking more into this relationship between plants that can't fix nitrogen, like cereals, and seeing what you could do to engineer that relationship and give those plants a way to work with those soil organisms. And then the third way, an even more complicated way of really taking whatever kind of taking that process out of those soil microorganisms and actually making the plant being able to do it itself, which seems like an incredibly difficult process. People have challenges doing much simpler things in genetic engineering. So it's certainly not to be underestimated how complicated that is. But we are kind of making some strides in this. So I'm curious, kind of based on those three approaches. And you said the first approach is kind of already in play. People are kind of already working on these bacteria that share more. But kind of these three approaches, what do you think has the most what do you think has the most promise in the next kind of number of years for making the most progress? What's kind of exciting you as this field is developing, and as we're getting more tools available to us for genetic engineering, like CRISPR and so on?
Yeah, well, I think actually that most likely, and it's my opinion, I guess it is shared by others, that the synthetic symbiosis approach, where we're actually getting there's more sharing, if you like, between the microbes in the soil and the cereals, is likely to be the one that delivers in the short term. The other two projects I talked about, either creating the nodule symbiosis on the cereals or putting the nitrogen fixation genes into the plant, are more long term. It's difficult to know really. We have some issues in the sense that with the manipulating the inoculants, we're not necessarily, as I've said, there are these issues with making it reproducible in different areas of the world, and also the fact that it probably will not be as efficient as something like a nitrogen fixing plant or a cereal which has a real genuine symbiotic association. Nevertheless, if that technology would work well in sub-Saharan Africa, that would be tremendous, because even a small increment in nitrogen delivery to crops would be incredibly important for African farmers. So I think most of us think the synthetic symbiosis is going to deliver. And as I've said, it's already been used in the world anyway, and there are promising developments on this, and people are building in new varieties of bacteria. We can even make synthetic nitrogen-fixing bacteria by, for example, taking a bacterium that we know is very, very good at associating with the plant, and even engineering the nitrogen fixation genes into that organism. So there are a lot of opportunities, I think, initially in the next couple of years with the synthetic symbiosis idea. I think the issues that I've already mentioned are whether or not it can be reproducible from year to year, and if it is not, do we lose some farmer confidence as a consequence?
And there were 2 points that you made earlier that I want to revisit that I think are relevant for farmers right now. And that is one being the issue of kind of adding synthetic nitrogen to a field that already has this process going on, and that you said that kind of halts the process. So maybe we could talk a little bit about the need for, I guess, specific kind of timing of application of synthetic fertilizer and controlled rates of synthetic fertilizer, the implications for overusing that type of product, and what that means for these organisms in the soil that are helping us. So that's one point, and then the other point about the oxygen sensitivity, and I guess the oxygen sensitivity kind of comes into play when we're talking about no-till agriculture. Is that correct?
Not necessarily. Do you want to talk about oxygen first then?
Sure.
Oxygen is really very, very complicated, actually, because the actual conditions of oxygenation are varying all the time. So if, for example, you have heavy rain and everything is really seriously flooded, then, and now we're not talking about in the plant, we're talking about maybe associated with the roots. And then if you get a flooded soil, then the thing's going to go pretty anaerobic. And the trouble is with these organisms when they're out there in the soil is that all of them have somehow different requirements for oxygen. And what that means, I suppose, practically is that they have different oxygen sensitivities. So some are much more sensitive than others. So some will fix nitrogen at one oxygen tension and others will at a much lower one. But when you're shifting all that balance by, for example, flooding, or you have a sort of desiccation system, or you have a situation when you're changing the soil, if you like, environment by, as you say, tilling it or whatever, whatever you're doing there, the oxygen tension is going to vary quite a lot. once you get close to the plant, then things change quite a lot, because you're going to be on the root surface, and again, you may have alterations in oxygen tensions. When you get inside the plant as an endophyte, you're then having to deal with the amount of oxygen which is in there. So I think what I'm trying to say is that one of the problems here is that you won't always have the optimal oxygen concentration to support nitrogen fixation, and that will give you a lot of variability. So did you want me to talk about fertilizers?
Yeah, let's go into
the initial part of your question, I think.
So yeah, thank you for covering that bit and clearing that up for me about the oxygen. But yeah, now going into the synthetic fertilizer, I guess, how do you see the effect of overuse of synthetic fertilizers on the existing nitrogen fixing bacteria that are around, and how does that actually affect farmers?
Well, first of all, I think what we need to consider is that if we're just talking about, the survival of the planet, if you like, it's a double-edged sword. I mean, we have to feed the world, and really fertilizers have supported an increase in the growth of the world's population, together with the Green Revolution, if you like, since 1950. For example, where there was a population of 2.5 billion, and it's almost 8 billion today. And how has that increase been possible? And we know that 50% of the human population now fed as a consequence of farmers using chemical fertilizers. Now that's an amazing success story, if you like. So on the one hand, you have that. And on the other hand, you have these huge unintentional consequences of that. And we all know about that, about fertilizer being lost to the environment. We see that everywhere, terrestrial, aquatic, marine environments, wherever. You've seen these algal blooms due to fertilizer runoff. And everybody's heard about nitrate contamination in drinking water, for example. And then you have all these transformations which are going on when you add fertilizers to soil and so on. You get huge transitions in the soil, and you then get all this atmospheric release of volatilized nitrogen, various oxides of nitrogen. For example, nitrous oxide is a gas which is 300 times more potent than carbon dioxide as a greenhouse gas. Well, luckily it's not as abundant as carbon dioxide. And it also depletes the ozone layer. So nitrogen fertilizers have a huge environmental problem. And as I've said, farmers in the past, because they know that they're so good at delivering yield, even throw them on legumes, which makes no sense at all. But it's a question of realizing that and nurturing nitrogen fixation and realizing the effect that fertilizers have on nitrogen fixation. But as I've already said, it's a balance. The farmer wants yield. That's what they're interested in. If you cut yield, then you're in trouble. And if you cut nitrogen, you will have trouble unless you can supplement it with biological nitrogen fixation. Now, there are other things that people are trying, which are also important. So nitrogen fixation, I should say, is not the only way to solve some of these issues. And there are a large number of scientists working on what they call nitrogen utilization efficiency, or NUE, sorry, nitrogen usage efficiency. So what that means is that you try to find ways of reducing the amount of nitrogen. And for example, you use, for example, slow release materials. So they're embedded in the right kind of environment, which allows only slow release of ammonia for the use of the plant. You can also think about engineering the plants for better nitrogen use efficiency, and we're learning a lot about that. So you can decrease your application of nitrogen fertilizers to a certain extent and still have quite good yields. So I think this combination of working on biological nitrogen fixation with also this kind of NUE approach will help us to drop down the amount of chemical fertilizer we're currently using. So we still may be I think ultimately it's going to be very difficult in the future, but we are going to be able to sustain the world's population and do something for sustainable agriculture. But I think we're going to have to develop this combination of work on biological nitrogen fixation and nitrogen usage.
Thank you, thank you. So if you're kind of just tuning into us, we've been speaking with Professor Ray Dixon from the John Innes Center in the United Kingdom. We've been talking about engineering of biological nitrogen fixation. Professor Dixon, as we wrap up this conversation, we're kind of nearing the end of the hour. I'd like to know if you could tell us a little bit more about the graduate students who are working with you, the different scientists who are working with you at the John Innes Center, and what are they working on, and what is kind of exciting you about your colleagues' work, and if there are any kind of recent publications or kind of upcoming developments that you would like our listeners to know about if they want to learn more about this subject.
That's quite a lot of questions, but I'll try and start with the first one. And maybe it's not answering it entirely, but I think, as I've already sort of tried to explain, solving this crisis is a huge challenge. And I'm not doing it on my own. I couldn't possibly. And my lab could not possibly attempt this. And it's really benefiting from a huge multidisciplinary collaboration between scientists worldwide. And from my lab is collaborating with colleagues in Brazil, across the UK and the United States, across Europe. I have very important collaborations with colleagues in China. And so science is global. We can't do this alone. Some have likened this challenge to a Manhattan Project in the sense it requires concerted long-term funding and collaboration on a huge scale. And we have all these three alternative approaches, but we've got a long way to go. And I should say that this science is benefiting from philanthropy in the sense that the Bill and Melinda Gates Foundation are funding some of these projects. In terms of what we're doing in my lab, specifically in my lab, we're working on the synthetic symbiosis projects and also engineering the nitrogen fixation genes into plants. So we have recent publications on actually giving the plant a bit of food from the bacteria, the release of ammonia. And we have that engineered so it's dependent on certain types of carbon source that can be provided by the root exudates of plants. So that's the the first project that we're working on. And we're also working on this project of engineering the genes into plants. And that's moving along in a kind of incremental way. We end up sometimes with challenges, or we find sometimes we don't need to use the bacterial genes. There's some genes in there in the plant that we can use. They're not nitrogen fixation genes, but they support the process. So we thought we'd have to use some bacterial genes to support it, but there are some plants that will do, plant proteins that will do the job. So that's one thing that we've been working on, and it's technical, but those things are called electron transfer proteins, electron transfer complexes. And as I mentioned, we always come across bottlenecks in this kind of work, and we had one of the components of nitrogenase which got cleaved when it went into the mitochondria. And so we had to ask the question, why is this happening? Why is this protein being degraded? Then we use synthetic biology to actually modify the nitrogenase so it's stable in the plant. So those are the kind of things that we're dealing with. If people want to read about this stuff, I mean, I think there are lots of reviews on this. Most of them, unfortunately, are quite technical, but I'm very pleased to sort of add some suggestions for reviews that people might like to read.
Yeah. Did you have more to add?
No, not really. No,
Okay.
that's where I...
Okay. I think that kind of brings us to a close. I just want to again thank you so much for your time this afternoon, Professor Dixon. So this has been Cornell Alliance for Science Live. The Alliance for Science is based at Cornell University in the Department of Global Development. We seek to promote access to scientific innovation as a means of enhancing food security, improving environmental sustainability, and raising the quality of life globally. You can follow us on Twitter at Science Ally, and we're also on Facebook at Science Ally. Alliance for Science Live will be syndicated on Google and Apple Podcasts and also Spotify. So you can subscribe to us there and also check out recent episodes. And we also want to congratulate our director, Sarah Evangia, on the recent, she won the CAST Award for Science Communication, which was just announced earlier this week. So we want to congratulate her on that. And once again, we've been speaking with Ray Dixon this morning and afternoon on his side. Thank you very much for your time today, Professor Dixon.
It's been a pleasure to talk to you, Chris. It really has. I've really enjoyed it.
Yeah, great. I hope we can have you on again another time soon, and I wish you all the best in your ongoing research on this subject, in the dream of sustainable agriculture with biological nitrogen fixation. So thank you.
Thank you, Chris.